the embryogenesis of the ear. And this isn't just about hearing. It's about a single integrated structure that's responsible for both our connection to the sound world and our internal sense of gravity and balance.
And when we look at the source material, all the detailed embryological research and molecular patterning studies, what immediately jumps out is that this incredible system is not built as one cohesive unit.
No, not at all. It's actually comprised of 3 entirely separate yet perfectly synchronized developmental systems.
Huh.
You've got the inner, the middle, and the external ear.
That's the core paradox we're tackling, isn't it? Our goal is to follow this blueprint chronologically, stage by stage, starting with the earliest molecular instructions and tracing the journey all the way through.
All the way through to the anatomical construction, and even that critical period of postnatal maturation. We really need to unlock the specific molecular and anatomical steps required to build this sensory marvel.
The foundation of this development is, it's immediately complex. It's a process that demands highly coordinated tissue interaction. It relies on a specific orchestra of genetic patterning signals and transcription factors. And it's not just the developing ear tissue itself that's involved.
Not by a long shot. Think of it as a highly complex construction project where the site manager needs flawless communication between, say, 5 different specialized teams all at once.
Okay, so who are the 5 teams?
Well, you have the precursor inner ear tissue itself. Then there's the surrounding mesenchyme, which will later become bone and cartilage. You have the adjacent hindbrain, which provides crucial neural timing signals.
And the neural crest cells, right?
Exactly, the neural crest cells, which are sort of the nomadic stem cells of the developing embryo. And then you even have signaling cues coming from the notochord, that central rod that defines the body axis.
Wow, so the slightest failure in coordination among any one of those 5 foundational structures?
And the entire sensory system faces catastrophic failure. That's the level of precision we're talking about, right from stage 1.
Okay, let's unpack this blueprint chronologically then. Starting with the foundation, the inner ear, or the labyrinth. This is the heart of sensory reception. And its formation is, it's a sequential process of astonishing speed and precision. When does this whole journey kick off?
It begins remarkably early. The very first signs of inner ear development appear shortly after the initial indicators for the developing eyes, which puts us around embryonic stage 9.
Stage 9. So the blueprint for hearing is already being laid down before many other major systems are even properly established.
That's right. And what this first sign looks like is something called the otic placode. If you were to look at the embryo's surface, you'd see 2 patches of ectodermal thickening, basically. Surface skin cells that have received a molecular signal saying, okay, your job is to form a deep sensory structure.
And their location is very specific, I gather.
Extremely specific. They're located laterally to the developing hindbrain, and the source is pinpointed to be lateral to rhombomeres 5 and 6 and just dorsal to the second pharyngeal cleft.
That precise location lateral to rhombomeres 5 and 6, that's crucial, isn't it? It's not just a random spot.
Oh, absolutely not. It's a perfect example of what you'd call developmental geography. That placement ensures the future otic structure is directly adjacent to the developing brain regions that will become the primary processing centers for hearing and balance signals.
So the structure and its eventual nervous connection are established simultaneously.
They're planned together from the very beginning.
So we have this thickening, the placode. How does this 2-dimensional patch of surface cells turn itself into a 3-dimensional fluid-filled organ?
It initiates a really fundamental process called invagination. The placode just sinks inward, forming what's called an otic pit.
A pit, like pushing your finger into a soft balloon.
That's a great analogy. Then, during stage 12, which is a time window from roughly 10 to 32 days post-fertilization, the pit completes its transformation. It actually pinches itself off entirely from the surface ectoderm.
And the result of that separation, what's left behind?
What remains is a simple hollow epithelial sac. This is the otic vesicle, sometimes called the otocyst or auditory vesicle. This tiny, sealed, fluid-filled balloon, now protected beneath the surface, is the absolute genesis point for the entire membranous labyrinth.
Okay, so once we have this simple otic vesicle, it's not just a generic balloon anymore. It has to immediately begin differentiating based on its future functions, hearing versus balance.
Exactly. The vesicle quickly massed itself out into what embryologists call prosensory domains. And these aren't just random regions. They're pre-programmed to give rise to 2 massive components.
Which are?
First, the entire membranous labyrinth, all the fluid-filled tubes, ducts, and chambers. And second, the vestibulocochlear ganglia, which will form the 8th cranial nerve, the dedicated neural superhighway connecting the ear to the brain.
And we see the first physical evidence of this specialization around stage 14, right? about 32 to 34 days post-fertilization. The initial, simple, pear-shaped identity of the vesicle is quickly lost.
It is. The first morphological change defines the future dorsal-ventral axis. The balance structures are going to be dorsal, and the hearing structure will be ventral. From the dorsomedial rim of the vesicle, a little tubular diverticulum begins to grow out, the endolymphatic appendage.
And the rest of the vesicle splits.
Yes, the remainder differentiates into two main regions, an expanded pars superior up top and a narrower pars inferior down below. This simple division sets the structural stage for the balance mechanism in the superior part and the hearing mechanism in the inferior part.
The endolymphatic appendage sounds important. It's not just a random offshoot.
No, it is absolutely essential for long-term function. That appendage elongates quite a bit, and its tip expands into what becomes the endolymphatic sac.
And it stays connected.
It stays connected back to the pars superior by a narrow endolymphatic duct. This entire apparatus is what will later regulate the volume and pressure of the endolymphatic fluid. I mean, if this appendage develops incorrectly, you can have conditions like Meniere's disease later in life.
Wow. So decisions made in the first month of development can directly impact adult health decades later.
That's the story of embryology in a nutshell.
So the next phase is sculpting the structures for detecting angular acceleration, the 3 semicircular canals. And this involves a really fascinating process of building and then selectively dissolving tissue.
It's an almost subtractive artistic process. It's quite beautiful. These canals emerge from that dorsal pars superior as two plate-like diverticula, one vertical, one horizontal. The epithelia in the center of these outgrowths then fuse together, they coalesce, to form something called a fusion plate.
Wait, hold on. So the structure grows outward as a solid sheet, and then the center of the sheet is deliberately killed off. That seems kind of counterintuitive for construction.
It does, but it's genetically programmed precision. The central part of that fusion plate is eventually resorbed or dissolved through programmed cell death apoptosis.
And what's left behind is the donut shape of the canal itself.
Exactly. The remaining space is perfectly tailored to measure fluid movement. The vertical plate gives rise to the anterior and posterior semicircular canals, which actually share a single common attachment point to the main chamber. And the horizontal plate becomes the lateral semicircular canal.
And the small expansion where the sensory hair cells will eventually reside, the ampulla that forms at one end of each of these canals, the whole thing works like a tiny, highly sensitive, liquid-filled gyroscope, doesn't it?
It's constantly reporting on three-dimensional head movement. And at the same time this is happening, the central chamber, the utriculosaccular chamber, is also differentiating. It's dividing into the structures for static balance.
So that's the utricle and the saccule.
Right. It divides into a smaller ventral saccule and a larger dorsal utricle. This division is achieved mainly by a horizontal infolding that extends toward the opening of that endolymphatic duct we mentioned earlier.
And that infolding is key because it restricts communication between the utricle and saccule to that narrow utriculosaccular duct, so they can function as separate but connected sensory organs.
Exactly. And as all this internal sculpting is happening, the entire membranous labyrinth undergoes a critical spatial reorientation. Its long axis, which was originally vertical within the early embryo's head, rotates.
It rotates to become horizontal.
More or less horizontal, yes. This ensures the vestibular organs are positioned correctly to measure gravity and linear acceleration in relation to the ground. It's an incredible bit of choreography.
All right, now we shift focus to the ventral apparatus, the pars inferior, which is destined to become the coiled receiver for sound, the cochlea.
Right, so around stage 15, the ventral tip of this pars inferior begins its remarkable elongation. This starts as immediately directed evagination, the cochlear elongation, which is evident in the ventral part of the chamber.
And this coiling is essential for frequency discrimination, right? The length difference allows high frequencies to be sensed near the base and low frequencies near the apex. How precise is this coiling mechanism?
It is a remarkable feat of growth and geometry. The proximal region just keeps increasing in length, and the distal region progressively coils around a central axis. The sources are very clear that it reaches its final configuration of 2.5 turns by post-fertilization week 10.
2.5 turns. Is that number significant?
It's hugely significant.
Yeah.
That 2.5 turn spiral is unique to humans and many primates, and its precision is absolutely vital for the functional range of human hearing. If you end up with a reduced number of turns, say 1.5, a condition known as Mondini dysplasia, the listener will suffer from profound frequency discrimination deficits.
So once that duct reaches its final length and spiral shape, the connection point back to the balance system has to be maintained but restricted.
Correct. The proximal part constricts, forming the ductus reunions. This narrow duct ensures the saccule maintains a fluid connection to the cochlea, which maintains the continuity of the entire endolymphatic system, uniting both balance and hearing functions.
And this is where the molecular blueprint has to shift from broad anatomical shaping to hyperfine cellular patterning. The central domain of the cochlear duct has to be patterned to give rise to the functional unit of hearing, the organ of Corti.
This is truly molecular engineering at its finest. To define the organ of Corti, you need incredibly precise regulatory signaling gradients. We know from the research that specific molecular pathways, things like bone morphogenetic protein or BMP, fibroblast growth factor, FGF, and Notch signaling are all absolutely required.
So how do these signals work to arrange the, 16,000 or so hair cells in such an intricate pattern?
They establish defined spatial territories. BMP and FGF signaling patterns are key to defining the sensory territories. These are the small strips where the hair cells will actually develop, versus the vastly larger non-sensory territories, which become support structures and fluid-modulating cells.
And Notch signaling, what does that do?
Notch acts as a sort of lateral inhibition pathway. It ensures that once a cell commits to becoming a sensory hair cell, it actively stops its immediate neighbors from doing the same thing.
Ah, so it creates spacing.
It creates the precise, organized rows of one inner hair cell and 3 rows of outer hair cells that are necessary for acoustic amplification and detection. Without that fine-tuning, if the signaling is too broad or too weak, you just get a disorganized mess. The result is profound deafness, even if the physical coil looks perfectly fine.
The cell lineage of the inner ear is one of the most surprising twists in this story. Let's clarify who builds what. What are the specific cell types that come directly from that early otocyst?
Okay, so the autocyst derivatives include the structural components of the tubes and chambers. That's things like the sustentacular cells, the support cells, the bipolar neurons that populate the vestibular and cochlear ganglia, and the unique epithelia that produce the endolymph.
So they form the housing, the fluid control, and the primary wiring.
Basically, yes, but here is the critical nuance that defines the function. The actual mechanosensory hair cells themselves, the ones in the organ of Corti, the maculae, and the cristae, they are not derived from the otocyst.
Wait, really? Where do they come from then?
That's the surprise. The hair cells actually originate from the placode cells, those surface ectoderm cells that were committed way back at the beginning. They merge into the structure and form these highly intricate mosaic patterns to create the sensory patches. The surrounding otocyst guides them and houses them, but the sensory machinery itself comes from that initial surface commitment.
So the very essence of sensing sound and balance originates from that earliest surface thickening. Is there anything else that doesn't come from the otocyst or the placode?
Yes, there is one and only one exception to this rule within the sensory inner ear, and it highlights the complexity of tissue migration. The melanocytes, the pigment-producing cells found in the stria vascularis.
Which is vital for regulating the fluid.
Vital for endolymph production and ionic regulation, yes. Those cells are derived from the neural crest. This is a tiny but crucial contribution from the neural crest to an otherwise placode-driven system.
So the variation in the final sensory structure, the flat maculae for gravity versus the complex structure of the organ of Corti for frequency, that must be a result of the molecular instructions acting on these early placodal cells.
Precisely. The different morphologies just reflect differing spatial and temporal expression of those morphochimicals like BMP and Notch during maturation. What's even more precise is the development of hair cell planar polarity.
Which is what, exactly?
It's the process where all the tiny hairs on the hair cells, the stereocilia, must orient themselves in the exact same direction. This development happens concurrently with the formation of the macule and saccule, ensuring that the movement of fluid can be converted into a directional neural signal. If that polarity is lost, the hair cell might still detect a sound, but the brain can't interpret the direction or intensity accurately.
Let's follow the neural path. We need the 8th cranial nerve, the vestibulocochlear ganglion, to transmit all this information to the brain.
Right. The rudiment for the ganglion appears at stage 13. Neuroblasts, those placode-specified cells, they delaminate from the anteroventral region of the otic vesicle. They proliferate, they migrate, and they coalesce to form the ganglion structure.
And you mentioned a unique fact about their origin.
I did. They're unusual because they're exclusively placodal in origin. This is a critical distinction. Most other cranial ganglia, like the trigeminal, have a dual origin from both the placode and the neural crest.
So why is the 8th nerve different?
Well, being purely placodal, suggests a potentially ancient or a highly isolated developmental lineage. It requires no neural crest contribution for its afferent function, which is relaying balance and auditory information to the brain. These neurons also become enveloped in thin myelin sheaths very early on, which is necessary for rapid signal transmission.
So that covers afferent transmission sound and balance going to the brain. But we know the brain actively controls and dampens hearing. How does that control system develop?
That is the efferent innervation, the olivocochlear bundle. This is an outgrowth of axons that originate from neurons located deep within the pons of the hindbrain in the superior olivary complexes.
And they grow out to the ear.
They grow out, travel alongside the eighth nerve, and provide efferent innervation, primarily terminating on the outer hair cells within the organ of Corti. This efferent system acts as a sophisticated gain control, allowing the brain to modulate the sensitivity of the ear to protect it from loud sounds and to help it focus on specific inputs.
Finally, what does this highly developed system actually hear before birth? The middle and external ears aren't fully air-filled yet.
That's a great question. The environment inside the uterus is acoustically rich. You've got the mother's heartbeat, digestive sounds, her voice, but it's a fluid medium. The sources confirm that in utero, the fetus receives sound not through the air conduction mechanism we use postnatally, but predominantly via bone conduction.
So vibrations travel through the skull.
Exactly. The developing skull bones transmit vibrations directly to the fluid-filled inner ear, confirming that the sensory function is active long before birth.
With the delicate membranous labyrinth now fully sculpted, it needs a protective shell, the bone. This process involves the surrounding mesenchymal cells undergoing chondrogenesis.
Right. This mesenchyme forms the cartilaginous otic capsule, which acts as a rigid frame. This happens between postmenstrual weeks 16 and 23. It's the temporary skeleton for the bony labyrinth.
And this frame must then ossify to become one of the densest, most protected bones in the entire body, the petrous part of the temporal bone.
Indeed. The capsule ossifies through a process called endochondral ossification to form almost the entire bony labyrinth. But there's a variation in the mechanism here that's really interesting.
Oh.
Specific structures like the modiolus, that's the central pillar of the cochlea, and the ossispiral lamina, they ossify directly from connective tissue. It's a process called intramembranous ossification, and it bypasses the cartilage stage entirely.
So once the bone is forming, we need the hydraulic cushion that protects the membranous labyrinth. We've talked about endolymph inside the labyrinth. Now we need perilymph.
Perilymph fills the vacuoles that develop in the connective tissue space between the newly formed cartilaginous capsule and the epithelial wall of the membranous labyrinth. Functionally, perilymph is chemically a lot like cerebrospinal fluid, and it acts as a hydraulic shock absorber.
We see the first development of these spaces quite early on.
We do. The rudiment of the periodic cistern, or the vestibular perilymphatic space, is visible in the reticulum between the saccule and the fenestra vestibuli by stage 17. This sets up that critical interface with the middle ear.
And this leads to the development of the cochlear fluid spaces, the scala vestibuli and the scala tympani.
Exactly. These spaces develop by the fusion of smaller vacuoles, though the precise cellular mechanism is still an area of active research, and they gradually extend all the way along the sides of the cochlear duct. And crucially, they communicate at the very apex of the coiled duct through a narrow passage.
The helicotrema.
Helicotrema, yes.
Yeah.
And that connection is vital. It ensures that the pressure wave entering the scala vestibuli at the base can dissipate correctly into the scala tympani, vibrating the basilar membrane along the way to detect frequencies.
Okay, we've successfully built the highly sophisticated receiver, the inner ear, deep within the temporal bone. But a fluid-filled receiver can't efficiently capture sound energy traveling through air. It needs an impedance matching mechanism, and that's the middle ear.
That's the perfect conceptual bridge. The middle ear's main function is to amplify air vibrations and efficiently transfer that energy into the dense fluid medium of the inner ear. And the key to understanding this whole section is recognizing the common origin. All components of the middle and external ear develop from the first and second pharyngeal arches.
Let's start with the cavity and the pharyngotympanic tube, or the eustachian tube. These structures, responsible for pressure equalization, initially form as bilateral hollow extensions of the first pharyngeal pouches. They're called the tubotympanic recesses.
Right, and you can see those around stage 13. The medial part of that recess stays narrow and forms the pharyngotympanic tube connecting the developing ear to the pharynx. It's a ventilation system for the middle ear.
But the lateral part widens significantly.
It undergoes massive widening to become the early tympanic cavity, and the spatial relationships change dramatically as the inner ear structure swells. Initially, the recess lies inferolateral to the cartilaginous otic capsule.
But gets pushed around.
Well, as the otic capsule, that rigid shell enlarges, the tympanic cavity is displaced and shifts into an anterolateral position. It's not a passive displacement, though. A key supporting structure, the tegmen tympani, grows as a cartilaginous process from the otic capsule itself, and it locks the middle ear into its final protected location.
And this process curves caudally, defining the lateral wall of the tube and incorporating the cavity into the petrous region of the temporal bone.
Exactly. The whole middle ear is essentially walled off and protected by the growing skull base. And this structural expansion continues deep into gestation. The mastoid antrum, a key dorsal expansion of the cavity, appears quite late during the late second and early third trimester.
And it's only visible on ultrasound around week 34.
That's right. And this late development is why infants are particularly susceptible to mastoid infections. The region is still developing its bony partitioning.
Now for the three tiny bones, the malleus, inlinicus, and stapes, the physical links in the transmission chain. And they are all derived from those developmental superheroes, the neural crest cells.
It's a classic example of neural crest migration, defining the architecture of the head. The ossicles arise from crest cells that migrated specifically from rhombomeres 1 through 4 into the mesenchyme of the first and second pharyngeal arches.
Let's break down the origin by arch. The malleus and incus, the hammer and anvil, they derive from the dorsal end of the first arch, from Meckel's cartilage.
Yes, and this is a point of powerful conceptual significance. It links human embryology directly to evolutionary history. These two bones are homologous to the quadrate and articular bones found in the jaws of birds and reptiles.
Wow.
In fact, in suckling marsupials like a tiny joey, the malleus and incus are still temporarily used in jaw articulation before they migrate fully into their auditory role. This embryological fact confirms millions of years of evolutionary repurposing.
The sources also mentioned a fascinating detail, a transitory connection of the incus, but not the malleus, to the otic capsule.
That temporary tethering suggests an even closer initial developmental linkage between the transmission chain and the inner ear mechanism. It might help in its correct alignment during initial condensation. It's a fleeting moment of integration that quickly vanishes.
And the third bone, the stapes, or stirrup, develops from the second arch.
Specifically from the proximal end of Reichert's cartilage, yes. And the stapes formation is arguably the most dynamic. Its characteristic stirrup shape is directly imposed by a transient structure, the stapedial artery.
So the bone forms around an artery.
Exactly. During stages 16 through 20, the stapes cartilage grows and elongates around this major artery. A ring of cartilage forms around the vessel, which then involutes and disappears around stage 21. The resulting hole in the stapes is a permanent fossil record of that temporary artery.
And its footplate, the interface with the inner ear, has a dual origin, which is crucial context.
Absolutely. While the arch component, the crura and the head, comes from the neural crest, the stapedial footplate, which sits in the fenestra vestibuli, forms from cells within the otic capsule derived from paraxial mesenchyme.
Which is a completely different cell lineage.
Completely different. Paraxial mesenchyme is typically what forms somites and vertebrae. This dual origin highlights the necessity of integrating the ossicle assembly with the rigid, non-moving structure of the inner ear capsule. If that footplate alignment is off by even a micron, sound transmission is severely hampered.
Before the ossicles can function, they need stability and protection from acoustic shock. That's the job of the two tiny middle ear muscles.
Right. The end of the stapedius muscle appear near the stapedial artery and facial nerve in the second arch mesenchyme around stage 18. This muscle, innervated by the facial nerve, is responsible for the acoustic reflex, dampening loud sounds.
And the tensor tympani.
Almost simultaneously, the tensor tympani appears in the first arch mesenchyme, attaching near the malleus, and it's innervated by the trigeminal nerve.
Okay, now for the process of creating the air-filled space itself, cavitation. We started with a hollow recess, but the space around the ossicles is initially filled with mesenchymal tissue.
Yes, the early middle ear cavity is basically a packed container of neural crest mesenchymes. This mesenchyme acts as a temporary scaffold, but it must then retract or be resorbed to create the true air-filled tympanic cavity.
And this resorption is essential for the ossicles to swing freely and function as levers.
If this process fails, you end up with congenital ossicular fixation and conductive hearing loss. It's a critical step.
And the lining of this new cavity?
The inner lining, the hypotympanum, is primarily endodermal epithelium from that original pharyngeal pouch. However, there's some interesting experimental evidence from mice that suggests the epithelial lining might partially derive from neural crest cells that underwent a mesenchymal to epithelial transition.
So they can switch jobs, essentially.
Just shows how versatile these cells are in shaping the final anatomy.
How long do the ossicles stay embedded in this mesenchyme before they are finally free to move?
Quite late. Although they differentiate into cartilage starting in stage 18, they remain embedded in the mesenchymal roof of the tympanic cavity until postmenstrual weeks 29 to 32.
That's well into the third trimester.
It is. Only when the mesenchyme is fully resorbed do they become suspended by ligaments and covered in the middle ear mucosa as the cavity expands. This makes them the smallest bones in the body, fully formed to adult dimensions long before birth.
But the middle ear cavity itself is not fully mature at birth, right?
That's a key clinical takeaway. The human tympanic cavity is quite small and often contains residual fluid and mesenchymal remnants at birth. Full maturation, including proper aeration and development of the mastoid air cells, requires substantial postnatal growth.
Which is why infants are so prone to middle ear infections.
Exactly.
Moving outward, we have the external ear, the auricle or pinna, and the external acoustic meatus, the EAM. These are the collector structures designed to funnel sound energy efficiently toward the tympanic membrane.
And the development of the EAM, the ear canal, has actually seen a bit of a conceptual update based on recent studies. Traditionally, it was thought to develop entirely within the first pharyngeal cleft, which you can see from stage 12.
And the newer research suggests
Gene expression studies suggest a more rostral origin from the actual invagination of the first arch itself. But regardless of the precise starting surface, the functional development involves layers of ectoderm growing inward.
and by the end of the embryonic period, the surface ectoderm forms two layers.
Yes, the basal germinative layer, which becomes the epidermis, and the superficial layer, the periderm. And what's particularly interesting about the periderm is that it grows by mitotic activity independently of the developing dermis or the basal layer. It acts as a protective, almost temporary epithelial surface.
And counterintuitively, the ear canal does not start as a tube. It's a solid plug.
That's right. It begins as a solid epidermal meatal plate, which extends inward toward the developing tympanic ring, a ring-shaped cartilaginous support structure. The underlying mesenchyme forms the final cartilaginous part of the canal, while the epithelial plate is the precursor to the opening itself.
So if it's a solid plug, it must undergo canalization. How does that opening process work?
The cells within that meatal plate begin to separate and break down, starting around postmenstrual weeks 13 to 14. An open canal is fully formed by postmenstrual week 18, and this process leaves behind a migratory epithelium that continues to function postnatally.
And this postnatal function is the self-cleaning mechanism.
Exactly. The process of ongoing epidermal mitosis generates movement of keratinous debris earwax and sheds skin outward toward the exterior. It's an innate, self-cleaning biological conveyor belt, which is crucial for maintaining acoustic clarity.
And like the middle ear, the bony structure is still developing at birth.
Yes, the osseous part of the EAM, the final rigid bony framework, develops postnatally from the tympanic ring. This is why the external ear canal of a newborn is short and primarily cartilaginous, making it highly flexible and easily collapsed.
The tympanic membrane is the interface, the perfect barrier and the initial vibratory surface. Its structure elegantly summarizes the integrated origins of the ear.
It is the ultimate example of a tissue meeting point, a classic trilaminar structure, with each layer derived from a different major embryonic component we've already discussed.
Let's walk through them.
Okay, the outer layer is ectoderm, derived from that meatal plate. The inner layer is endoderm, derived from the tubotympanic recess of the first pharyngeal pouch. And critically, the core structural layer sandwiched between them is mesenchyme, derived from the neural crest.
So the eardrum is physically composed of tissue from all three major ear segments.
It is the functional handshake between the collector, the transmitter, and the protector. This membrane is supported by the tympanic ring, which later ossifies. Ossification centers for the surrounding bone appear around postmenstrual week 11, but complete development of the supporting bone doesn't happen until around 9 years of age.
That detail is vital for clinicians dealing with pediatric patients.
It is. Remember, the tympanic membrane and the ossicles have reached adult size in the neonate, achieving maximum acoustic efficiency early. However, because the bony EAM is still forming postnatally, the canal is very small and relatively soft.
Which makes looking inside a baby's ear very different.
It makes a coscopy visual examination of the eardrum a completely different clinical experience in infants compared to adults.
Finally, the visible external collector, the auricle or pinna. This structure is essential for sound localization.
Its development begins around stage 15, visible as mesenchymal proliferations beneath the ectoderm at the caudal border of the first pharyngeal arch and the cranial border of the second arch.
These proliferations were historically termed the auricular hillux, numbered 1 through 6. But this traditional view has been significantly refined, hasn't it?
It has, yeah. Modern 3D reconstructions and lineage tracing studies have clarified the origin. They emphasize that the bulk of the auricle, so the helix, antihelix, and concha, arises overwhelmingly from second pharyngeal arch mesenchyme.
So the contribution from the first arch is much more restricted.
Much more. The first pharyngeal arch mesenchyme primarily contributes only to the tragus and the anterior external acoustic meatus. And this distinction is clinically essential. Errors in first arch development, often seen in conditions like Treacher-Collins syndrome, result in specific, recognizable anomalies in those anterior structures.
Whereas second arch failures affect the major cartilaginous bulk of the ear.
Exactly.
And what about the soft, pliable bottom part?
The lobule is the last part of the auricle to develop, finalizing the ear's structure. Its late appearance means it's often spared from anomalies that affect the cartilaginous framework earlier in gestation.
We have completed the structural journey. We've built the 3 interconnected segments relying on an incredibly tight molecular choreography. Now we have to look at what happens when even one note in that molecular orchestra is missed, and why this developmental timeline is so critical in early life.
The consequence of developmental failure is tragically common. Prelingual deafness is the most common congenital anomaly, affecting anywhere from 2 to 6 out of every 1,000 newborns. And when you investigate the causes, at least 2/3 of cases have a genetic basis.
And those genetic cases are split between non-syndromic deafness, which is usually sensorineural and accounts for about 70% of cases,
And syndromic deafness, where the hearing loss is part of a broader condition, which accounts for the remaining 30%.
The sheer number of genes involved is a testament to the system's complexity.
It is. We're talking about essential signaling molecules and transcription factors, TGFB1, BMP4, EYA1, and many G-protein-coupled receptor genes. A defect in EYA1, for example, which is critical for Audicot play code formation, can lead to severe inner ear malformations. It also impacts renal development, too.
Which illustrates the shared regulatory pathways.
Right. Can you detail what these gene failures look like structurally inside the inner ear?
Well, the outcomes are very specific dysplasias. We see labyrinthine dysplasias, malformations of the coiled structure. The classic example is the Mundini deformity, where the cochlea is reduced to a single or 1.5 turns instead of the required 2.5.
And that reduction severely limits the capacity for frequency analysis.
Exactly. We also see things like an enlarged endolymphatic duct, which disrupts fluid homeostasis. And on the cellular level,
Microscopically, the molecular failures manifest as profound disorganization of the sensory machinery. The stereocilia, the tiny sensory hairs, lose their precise organization, and critical cytoskeletal components that anchor those cells are lost. This results in the loss of proper mechanoelectrical transduction.
we also noted functional failures related to electrochemical gradients.
Yes, this involves defects in K+, potassium ion, recycling across the gap junctions in the cochlear supporting cells. K+ is the key ion that depolarizes the hair cells, allowing them to fire. If the surrounding support cells can't recycle this K+ efficiently, the hair cells experience an energy crisis. They can't recharge and maintain the electrochemical gradient needed to transduce sound.
So you get profound sensorineural hearing loss even in a structurally perfect-looking ear.
Precisely. And it all emphasizes the interconnected nature of the system. Structural anomalies in the middle and external ear-like failure of cavitation or canal atresia are rarely isolated. These physical blockages prevent sound from reaching the inner ear, compounding the challenge.
Given that the auditory system is functional in utero from around postmenstrual week 20, early detection of hearing loss is absolutely paramount.
It is an urgent public health priority. Permanent hearing loss is significantly more common in high-risk groups, particularly preterm infants, with rates up to 30 per 1,000. And crucially, even preterms who pass the screening may have altered postnatal auditory processing because of their different early environmental exposure.
For the listener, understanding the 2 routine neonatal screening tools is crucial. They test different parts of the auditory chain.
The first, and often the initial screening tool, is evoked otoacoustic emissions, or EOAE. This is a functional test that detects low-level sounds emitted back by the cochlea, specifically the outer hair cells, in response to a click stimulus.
So it's checking if the cochlear amplifier is working.
Exactly. If the outer hair cells are compromised, The EOAE test is typically failed.
And if the EOAE is failed or if the baby is high risk, we move on to the neural pathway check.
That's the automated auditory brainstem response, or AABR test. This test records the generation of electrical waves in the brainstem in response to stimuli. It checks the integrity of the neural highway. Is the signal successfully being transferred from the inner ear along the 8th cranial nerve and up to the brain?
And the timing of these screenings is non-negotiable because of the rapid pace of brain development.
It must be treated as a developmental emergency. Screening has to occur before postnatal week 4, and any infant who fails the screening must have comprehensive diagnostic evaluation completed before postnatal month 3. Missing this narrow window means delaying intervention, which has profound consequences for the developing brain.
This brings us to the climax of the story. The brain is not finished learning how to hear at birth. The loss of auditory input in those early months profoundly affects the postnatal patterning and maturation of the temporal cortex.
It's the neuroplasticity paradox. The structural components may be fully formed, but the cortex needs input to specialize. This phenomenon is directly analogous to the effect of visual deprivation leading to amblyopia. If the sensory highway is silent, the processing center adapts by reducing its capacity.
So the auditory cortical regions are there, but the wiring isn't complete.
The complex interconnections, the true processing networks, mature rapidly through sustained postnatal acoustic experience.
The functional skill that develops during this period is called auditory temporal resolution.
And temporal resolution is our brain's capacity to detect minute time intervals between sound stimuli. This seemingly abstract skill is absolutely foundational for human communication. It's essential for perceiving rhythm and, crucially, for discriminating the frequency and duration of phonemes.
Which are the smallest units of sound that differentiate meaning in language.
Exactly. The difference between pat and bat. If you cannot detect those tiny timing differences, language acquisition is severely delayed.
And this rapid specialization is demonstrated by how quickly the brain tunes itself to local language.
It is astonishing. Neonates can discriminate their mother's voice even before postnatal month 1. They are initially generalists, sensitive to all acoustic differences across all languages, but they rapidly specialize, becoming fine-tuned to the distinctive phonemes of their mother tongue. They actively shed sensitivity to sounds they don't hear often.
So if that specialized sound input is missing due to congenital deafness, this cortical specialization can't happen correctly.
And the consequences are not just poor hearing, they are severe developmental and neurological changes. You see delays in speech and language acquisition leading to severe learning difficulties. And we have neurological evidence of this physical change.
What does that look like?
Untreated congenital deafness leads to observable structural alterations in the brainstem, the cochlear nuclei and the inferior colliculi, and critically reduced white matter volume in the auditory cortex. The pathway literally atrophies or gets repurposed by other senses without adequate input.
Which is why the timing of intervention is everything.
Absolutely. Cochlear implants are the optimal therapy for prelingually deaf infants, but the intervention must occur within that developmental sweet spot, ideally within postnatal years 1 through 3. This window is necessary to allow the auditory cortex the time required to properly reorganize and acquire the necessary plasticity for functional language acquisition.
Implanting later provides sound awareness, but
it often fails to restore the brain's full capacity for complex language processing. The window has closed.
we have journeyed through the creation of a triple-layered sensory system, from the simple ectodermal thickening of the otic placode at stage 9 through the molecular engineering required to pattern 2.5 cochlear turns, the bizarre process of the stapes forming around a vanishing artery, and the critical separation required for the middle ear cavity to even exist.
The key insight here is the staggering precision and the disparate origins that must ultimately integrate seamlessly. The inner ear relies on placode cells and paraxial mesenchyme. The ossicles are neural crest. The external ear is derived from arches and clefts. Yet a failure in one system.
Whether it's the molecular balance of K+ recycling in the labyrinth or the simple failure of mesenchyme resorption in the middle ear,
it has immediate cascading consequences for the whole thing.
and the final, undeniable truth is that the structural blueprint for hearing may be complete when the baby is born, but the specialized function is entirely dependent on the acoustic environment immediately afterward. The ear is built, but the brain must be taught how to interpret its signals.
That is the core takeaway. The auditory cortex only matures and specializes through that critical early postnatal period of acoustic experience. If that input is altered or diminished, the brain fundamentally changes its processing capacity for life.
So we noted that the long-term subtle effects of early perinatal acoustic exposure beyond just deafness are not yet fully understood. Considering how quickly the temporal cortex specializes based on sound input, differentiating the mother's voice before month 1, here is our final provocative thought for you to explore. If the brain is so plastic and sensitive in those first few months, what are the long-term subtle implications of a modern, acoustically saturated but often degraded sound environment? Think constant low-level noise, inconsistent parental voices, or the pervasive presence of artificial audio on the development of auditory, temporal resolution, and language specialization in children. We're not talking about profound deafness, but the quality of the cortical landscape we are setting for the next generation.
We'll leave you to consider the soundscape of infancy. Until the next deep dive.